Xenopus, ʻzɛnəpəs, GK, Xi epsilon mu omicron sigma, xenos equals strange, pi omicron upsilon sigma, pous equals foot, commonly known as the clawed frog, is a genus of highly aquatic frogs native to sub-Saharan Africa. 20 species are currently described in the Xenopus genus. The two best-known species of this genus are Xenopus laevis and Xenopus tropicalis, which are commonly studied as model organisms for developmental biology, cell biology, toxicology, neuroscience, and for modeling human disease and birth defects. Characteristics. Description. All species of Xenopus have flattened, somewhat egg-shaped and streamlined bodies, and very slippery skin, because of a protective mucus covering. The frog's skin is smooth, but with a lateral line sensory organ that has a stitch-like appearance. The frogs are all excellent swimmers and have powerful, fully webbed toes, though the fingers lack webbing. 3 of the toes on each foot have conspicuous black claws. The frog's eyes are on top of the head, looking upwards. The pupils are circular. They have no movable eyelids, tongues. Rather, it is completely attached to the floor of the mouth, or eardrums. Similarly to Pippa, the common Suriname toad. Unlike most amphibians, they have no haptoglobin in their blood. Behavior. Xenopus species are entirely aquatic, though they have been observed migrating on land to nearby bodies of water during times of drought, or in heavy rain. They are usually found in lakes, rivers, swamps, potholes in streams, and man-made reservoirs. Adult frogs are usually both predators and scavengers, and since their tongues are unusable, the frogs use their small forelimbs to aid in the feeding process. Since they also lack vocal sacs, they make clicks, brief pulses of sound, underwater. Again similar to Pippa, males establish a hierarchy of social dominance in which primarily one male has the right to make the advertisement call. The females of many species produce a release call, and Xenopus laevis females produce an additional call when sexually receptive and soon to lay eggs. The Xenopus species are also active during the twilight, or crepuscular, hours. During breeding season, the males develop ridge-like nuptial pads, black in color, on their fingers to aid in grasping the female. The frog's mating embrace is inguinal, meaning the male grasps the female around her waist. Species. Model organism for biomedical research. Like many other anorans, they are often used in laboratory as research subjects. Xenopus embryos and eggs are a popular model system for a wide variety of biological studies. This animal is used because of its powerful combination of experimental tractability and close evolutionary relationship with humans, at least compared to many model organisms. Xenopus has long been an important tool for in vivo studies in molecular, cell, and developmental biology of vertebrate animals. However, the wide breadth of Xenopus research stems from the additional fact that cell-free extracts made from Xenopus are a premier in vitro system for studies of fundamental aspects of cell and molecular biology. Thus, Xenopus is the only vertebrate model system that allows for high-throughput in vivo analyses of gene function in high-throughput biochemistry. Furthermore, Xenopus oocytes are a leading system for studies of ion transport in channel physiology. Xenopus is also a unique system for analyses of genome evolution and whole genome duplication in vertebrates. As different Xenopus species form a ploidy series formed by interspecific hybridization, Xenbase is the model organism database (MOD) for both Xenopus laevis and Xenopus tropicalis. Investigation of human disease genes. All modes of Xenopus research, embryos, cell-free extracts, and oocytes, are commonly used in direct studies of human disease genes and to study the basic science underlying initiation and progression of cancer. Xenopus embryos for in vivo studies of human disease gene function. Xenopus embryos are large and easily manipulated, and moreover, thousands of embryos can be obtained in a single day. Indeed, Xenopus was the first vertebrate animal for which methods were developed to allow rapid analysis of gene function using misexpression by mRNA injection. Injection of mRNA in Xenopus that led to the cloning of interferon. Moreover, the use of morpholino antisense oligonucleotides for gene knockdowns in vertebrate embryos, which is now widely used, was first developed by Janet Heisman using Xenopus. In recent years, these approaches have played an important role in studies of human disease genes. The mechanism of action for several genes mutated in human cystic kidney disorders, e.g., nephronophthisis, have been extensively studied in Xenopus embryos, shedding new light on the link between these disorders, ciliogenesis and WNT signaling. Xenopus embryos have also provided a rapid test bed for validating newly discovered disease genes. For example, studies in Xenopus confirmed and elucidated the role of PYCR1 in cutis laxa with progeroid features. Transgenic Xenopus for studying transcriptional regulation of human disease genes. Xenopus embryos develop rapidly, so transgenesis in Xenopus is a rapid and effective method for analyzing genomic regulatory sequences. In a recent study, mutations in the SMAD7 locus were revealed to associate with human colorectal cancer. Interestingly, the mutations lay in conserved, but non-coding sequences, suggesting these mutations impacted the patterns of SMAD7 transcription. To test this hypothesis, the authors used Xenopus transgenesis, and revealed this genomic region drove expression of GFP in the hindgut. Moreover, transgenics made with the mutant version of this region displayed substantially less expression in the hindgut. Xenopus cell-free extracts for biochemical studies of proteins encoded by human disease genes. A unique advantage of the Xenopus system is that cytosolic extracts contain both soluble, cytoplasmic and nuclear proteins, including chromatin proteins. This is in contrast to cellular extracts prepared from somatic cells with already distinct cellular compartments. Xenopus egg extracts have provided numerous insights into the basic biology of cells with particular impact on cell division and the DNA transactions associated with it. See below. Studies in Xenopus egg extracts have also yielded critical insights into the mechanism of action of human disease genes associated with genetic instability and elevated cancer risk, such as ataxia telangiectasia, BRCA1 inherited breast and ovarian cancer, NBS1 breakage syndrome, RECQL4 Rothman-Thompson syndrome, CMYC oncogene and FANC proteins, Fanconi anemia. Xenopus oocytes for studies of gene expression and channel activity related to human disease. Yet another strength of Xenopus is the ability to rapidly and easily assay the activity of channel and transporter proteins using expression in oocytes. This application has also led to important insights into human disease, including studies related to trypanosome transmission, epilepsy with ataxia and sensorineural deafness, catastrophic cardiac arrhythmia, Long-Quartz syndrome, and megalencephalic leukoencephalopathy. Gene editing by the CRISPR-Cas system has recently been demonstrated in Xenopus tropicalis and Xenopus laevis. This technique is being used to screen the effects of human disease genes in Xenopus, and the system is sufficiently efficient to study the effects within the same embryos that have been manipulated. Investigation of fundamental biological processes. Signal transduction. Xenopus embryos and cell-free extracts are widely used for basic research in signal transduction. In just the last few years, Xenopus embryos have provided crucial insights into the mechanisms of TGF-beta and WNT signal transduction. For example, Xenopus embryos were used to identify the enzymes that control ubiquitination of SMAD4, and to demonstrate direct links between TGF-beta superfamily signaling pathways and other important networks, such as the MAP kinase pathway and the WNT pathway. Moreover, new methods using egg extracts revealed novel, important targets of the WNT GSK3 destruction complex. Cell division. Xenopus egg extracts have allowed the study of many complicated cellular events in vitro. Because egg cytosol can support successive cycling between mitosis and interphase in vitro, it has been critical to diverse studies of cell division. For example, the small GT-pase run was first found to regulate interphase nuclear transport. But Xenopus egg extracts revealed the critical role of RAN GT-pase in mitosis independent of its role in interphase nuclear transport. Similarly, the cell-free extracts were used to model nuclear envelope assembly from chromatin, revealing the function of RANKpase in regulating nuclear envelope reassembly after mitosis. More recently, using Xenopus egg extracts, it was possible to demonstrate the mitosis-specific function of the nuclear lamin B in regulating spindle morphogenesis and to identify new proteins that mediate kinetic reattachment to microtubules. Embryonic development. Xenopus embryos are widely used in developmental biology. A summary of recent advances made by Xenopus research in recent years would include epigenetics of cell fate specification and epigenome reference maps, microRNA in germ layer patterning in eye development, link between WNT signaling and telomerase, development of the vasculature, gut morphogenesis, contact inhibition and neural crest cell migration and the generation of neural crest from pluripotent blastula cells. DNA replication. Xenopus cell-free extracts also support the synchronous assembly and the activation of origins of DNA replication. They have been instrumental in characterizing the biochemical function of the pre-replicative complex, including MCM proteins. DNA damage response. Cell-free extracts have been instrumental to unravel the signaling pathways activated in response to DNA double-strand breaks, ATM, replication fork stalling, ATR, or DNA interstrand crosslinks, FA proteins and ATR. Notably, several mechanisms and components of these signal transduction pathways were first identified in Xenopus. Apoptosis. Xenopus oocytes provide a tractable model for biochemical studies of apoptosis. Recently, oocytes were used to study the biochemical mechanisms of caspases 2 activation. Importantly, this mechanism turns out to be conserved in mammals. Regenerative medicine. In recent years, tremendous interest in developmental biology has been stoked by the promise of regenerative medicine. Xenopus has played a role here as well. For example, expression of 7 transcription factors in pluripotent Xenopus cells rendered those cells able to develop into functional eyes when implanted into Xenopus embryos, providing potential insights into the repair of retinal degeneration, a damage. In a vastly different study, Xenopus embryos was used to study the effects of tissue tension on morphogenesis, an issue that will be critical for in vitro tissue engineering. Physiology. The directional beating of multiciliated cells is essential to development and homeostasis in the central nervous system, the airway, and the oviduct. Interestingly, the multiciliated cells of the Xenopus epidermis have recently been developed as the first in vivo testbed for live cell studies of such ciliated tissues, and these studies have provided important insights into the biomechanical and molecular control of directional beating small molecule screens to develop novel therapeutics. Because huge amounts of material are easily obtained, all modalities of Xenopus research are now being used for small molecule-based screens. Chemical genetics of vascular growth in Xenopus tadpoles. Given the important role of neovascularization in cancer progression, Xenopus embryos were recently used to identify new small molecules inhibitors of blood vessel growth. Notably, compounds identified in Xenopus were effective in mice. Notably, frog embryos figured prominently in a study that used evolutionary principles to identify a novel vascular disrupting agent that may have chemotherapeutic potential. That work was featured in the New York Times Science Times. In vivo testing of potential endocrine disruptors in transgenic Xenopus embryos. A high-throughput assay for thyroid disruption has recently been developed using transgenic Xenopus embryos. Small molecule screens in Xenopus egg extracts. Egg extracts provide ready analysis of molecular biological processes and can rapidly screened. This approach was used to identify novel inhibitors of proteasome-mediated protein degradation and DNA repair enzymes. Genetic studies. While Xenopus laevis is the most commonly used species for developmental biology studies, genetic studies, especially forward genetic studies, can be complicated by their pseudo-tetraploid genome. Xenopus tropicalis provides a simpler model for genetic studies. Having a diploid genome, gene expression knockdown techniques. The expression of genes can be reduced by a variety of means. For example, by using antisense oligonucleotides targeting specific mRNA molecules. DNA oligonucleotides complementary to specific mRNA molecules are often chemically modified to improve their stability in vivo. The chemical modifications used for this purpose include phosphorothioate, 2,O-methyl, morpholino, mea-phosphoramidate and deed-phosphoramidate. Morpholino oligonucleotides. Morpholino oligos are used in both X. laevis and X. tropicalis to probe the function of a protein by observing the results of eliminating the protein's activity. For example, a set of X. tropicalis genes has been screened in this fashion. Morpholino oligos, MOs, are short, antisense oligos made of modified nucleotides. MOs can knock down gene expression by inhibiting mRNA translation, blocking RNA splicing, or inhibiting miRNA activity and maturation. MOs have proven to be effective knockdown tools in developmental biology experiments and RNA blocking reagents for cells in culture. MOs do not degrade their RNA targets, but instead act via a steric blocking mechanism in a independent manner. They remain stable in cells and do not induce immune responses. Microinjection of MOs in early Xenopus embryos can suppress gene expression in a targeted manner. Like all antisense approaches, different MOs can have different efficacy and may cause off-target, non-specific effects. Often, several MOs need to be tested to find an effective target sequence. Rigorous controls are used to demonstrate specificity, including phenocopy of genetic mutation, verification of reduced protein by Western or immunostaining, mRNA rescue by adding back a mRNA immune to the MO, use of two different MOs, translation blocking and splice blocking, injection of control MOs. Xenbase provides a searchable catalog of over 2,000 MOs that have been specifically used in Xenopus research. The data is searchable via sequence, gene symbol, and various synonyms, as used in different publications. Xenbase maps the MOs to the latest Xenopus genomes in gBrowse, predicts off-target hits, and lists all Xenopus literature in which the Morpholino has been published. Xenbase _Xenopus laevis and tropicalis web resource.